welcome to our presentation. Today, Linda, Noshin, and I will be presenting on the effect of interaction between methotrexate and dihydrofolate reductase on DNA synthesis in L1210 cells in vitro. To begin, here's a brief introduction on DHFR. Dihydrofolate reductase, otherwise known as DHFR, is mainly responsible for nucleotide biosynthesis. More specifically, it catalyzes the reduction of folate and dihydrofolate to tetrahydrofolate. It is a highly conserved function in many organisms. It has also been a target for both antimicrobial and anti-cancer drugs. In this research paper, we will be observing how methotrexate, an anti-cancer drug, impacts DHFR activity. Now more on the structure of DHFR and MTX.
Methotrexate competitively inhibits DHFR as it prevents substrate binding. Methotrexate is a structural analog of folate and will bind to the active site of DHFR with high affinity. This prevents the binding of DHFR's natural substrate dihydrofolate. When DHFR is inhibited, production of tetrahydrofolate is inhibited. In the synthesis of thymidine, tetrahydrofolate donates a methyl group to deoxyuridine monophosphate to form deoxythymidine monophosphate. When de novo synthesis of thymidine is disrupted, DNA synthesis and replication are disrupted. Methotrexate works as an anti-cancer drug because this inhibition of nucleotide biosynthesis disrupts the replication and life cycle of cancer cells, ultimately leading to cell death.
Methotrexate is an immunosuppressive drug that can be used in the treatment of various cancers, including breast and neck cancers. This makes MTX an extremely significant drug in cancer research. This study is important because its purpose is to examine the role of free and bound MTX in antitumor action and in the inhibition of DNA synthesis. Additionally, the activity of DHFR in L1210 tumors in vitro is studied. MTX has been used as a high-dose therapy and has relative success. Therefore, this research aims to study its mechanisms of anti-tumor action. There have been earlier research on MTX, including two done by Bertino and Weichheiser in the 1960s. These researchers believed there was a stoichiometric inhibition of DHFR by MTX. After their publications, more recent work by Goldman, Fife, Margolis, and others in the early 1970s have proved that instead, extra-free MTX are necessary for maximal DNA synthesis inhibition. Even though much research has been performed, at the time of this research, the role of free MTX as in MTX which are not bound to intracellular sites in DHFR inhibition was not understood. Also, the role of DHFR-bound MTX was not understood either. Determining the purpose of these two roles was the main purpose of these experiments.
Now let's move to methods.
L1210 leukemia cells were grown and harvested from CDF female mice. The cells were isolated using a series of hypotonic lysis, centrifugation, and resuspension. Then the cells underwent viability assessment using type N blue exclusion to ensure that a good portion of the cells were viable. The viable cells were then incubated in a hot water bath, and the cell counts were regularly adjusted to maintain a constant amount of cells per millimeter. A stream of warm oxygen and carbon dioxide mixture was passed over the incubated cells right before the MTX was added. The new mixture was allowed 20 minutes to incubate so that MTX was able to properly bind DHFR. Then the mixture was centrifuged and the cell pellet resuspended with varying amounts of radio-labeled MTX. The cells were again incubated. In 5-minute intervals, the aliquots of the cell suspension were taken and cell pellet was isolated. A portion of the supernatant was saved to determine the extracellular MTX concentrations. Meanwhile, the pellet was washed and plated on a disc to dry in an overnight oven. A sterile analytic Mark III liquid scintillation counter was used to count hydrogen efficiency in the resuspended pellet.
Cells were pulsed with deoxyuridine to examine the effect of methotrexate on DNA synthesis. After 20 minutes of incubation with labeled deoxyuridine, efflux was permitted for 40 minutes. 50 microliters of labeled UDR was added to give a final deoxyuridine concentration of 0.1 millimolar. Aliquots were sampled at 5-minute intervals, and incubation was terminated using trichloroacetic acid. Comparison of the radioactivity recovered in the TCA precipitate and that incorporated into the DNA was determined by a perchlorate extraction method. In a second trial, researchers added cyclohexamide an inhibitor of protein synthesis to prevent de novo synthesis of DHFR. Cyclohexamide has been shown to inhibit translation elongation through binding to the E site of the 60S ribosomal unit and interfering with deacetylated tRNA.
We tried to find a relationship between DHFR activity at different methotrexate concentrations by studying the specific activity of DHFR using enzymatic study methods. For 3 micromolar MTX studies, cell pellets were obtained after efflux. For 0.05 micromolar MTX studies, cell pellets were obtained after uptake. The pellet was then suspended in a tris HCl buffer with a 1:4 dilution of intracellular volume. Cells were then lysed by alternate freezing and thawing, and then centrifuged. Complete recovery of all activity was confirmed after the 3rd freeze-thaw procedure. Using a spectrophotometric method, the supernatant was recovered for DHFR activity assay. The assay mixture had tris HCl buffer at a pH of 7, which is the intracellular pH, KCl, NADPH, and the enzyme extract. The reaction was initiated by adding DHF, which was prepared from folic acid. The enzyme activity was determined by measuring the decrease in absorbance at 340 nanometers using a spectrophotometer at 37 degrees Celsius. The relationship between intracellular DHFR and tightly bound intracellular MTX was determined by titrating the DHFR with purified MTX. These cell volumes were calculated by determining the cell wet and dry weights. Now let's move on to results.
Table 1 summarizes the total intracellular MTX content in each experimental condition, which uses different concentrations of MTX as depicted in the leftmost lane. Using a concentration of 0 MTX added, a non-exchangeable MTX fraction of 4.27 plus or minus 0.02 nanomole over grams, dry cell weight was determined. This value was then subtracted from each total intracellular MTX to determine the exchangeable intracellular MTX concentrations. The MTXI concentration in the right lane is the intracellular MTX concentrations. Figure 1 depicts the cellular MTX concentrations as a function of time in a medium with 0.05 micromolar extracellular MTX concentration. Since only 60 minutes were allowed for uptake, the high-affinity intracellular sites were not saturated. Therefore, you can see the graph as linear for the solid line. For the dashed line, cells were washed 2 times and placed in an MTX-free medium at 30 minutes. This is why the cellular MTX levels did not increase past that time.
Table 2 shows deoxyuridine incorporation under varying intracellular concentrations of methotrexate. Following uptake and a 40-minute efflux period, during which a steady state was reached, cell suspensions were pulsed with labeled deoxyuridine, and incorporation permitted for a 30-minute interval. It was observed that high concentrations of methotrexate did not alter the rate of UDR entry into cells. However, Table 2 shows that methotrexate concentration did alter the rate of deoxyuridine incorporation. We can see that a higher concentration of intracellular methotrexate is correlated with a lower percentage of deoxyuridine incorporation. Extracellular methotrexate concentrations in excess of 1 millimolar depressed UDR incorporation to within less than 2% of control. In another trial, addition of cyclohexamide, an inhibitor of protein synthesis, decreased the deoxyuridine incorporation rates. These rates were lower than the control and experimental conditions in which cyclohexamide was not present. This decrease may reflect secondary inhibition of DNA synthesis at high cyclohexamide concentrations. Table 3 shows DHFR activity at various intracellular methotrexate levels. Methotrexate concentration is expressed in terms of dry cell weight. DHFR activity is expressed as a percentage of a control. No DHFR activity could be detected at a methotrexate concentration greater than 1 millimolar. At a methotrexate concentration of 0.69 millimolar, DHFR activity began to recover. From that point, a low but increasing level of enzyme activity was detected with decreasing methotrexate concentration. When all intracellular sites are saturated and there is no free methotrexate, the activity was 6.09% of control. When methotrexate concentration is low, DHFR activity recovers to within 78% of control.
As listed out in Table 3 and shown in Figure 2, DHFR activity increases at lower concentrations of MTX. Figure 2 plots the UDR incorporation and DHFR activity against various total intracellular MTX contents. Each point represents the mean value of at least 3 performed experiments. The line with the black dots represent UDR incorporation, while the line with the white dots represents DHFR activity in terms of a percentage. The points to the right of the arrow represents the total intracellular MTX in excess of high affinity binding sites, while the points to the left of the arrow represents the opposite, or when there is a lack of total intracellular MTX compared to binding sites. The figure shows that DHFR enzyme activity increased to 78.7% when only 16.6% of the enzyme sites were saturated, assuming that there is a 1-to-1 ratio of MTX to DHFR. Similarly, UDR incorporation was also enhanced to 130% or more at these same points. Additionally, it is important to note that there is a sigmoidal relationship between total intracellular MTX and DHFR activity, or UDR incorporation rate. The curve is most compatible with a slowly reversible enzyme inhibitor interaction, where in most cases the inhibitor is enzyme bound. Thus, this suggests that the MTX/DHFR association is slowly reversible. Figure 3 is derived from Figure 2, and it illustrates the relationship between DHFR activity and UDR incorporation. Both figures demonstrate that the recovery of DNA synthesis is seen to occur concurrently with DHFR activity, but at a much slower rate. Maximal suppression of both DHFR activity and UDR incorporation appears to require D's free intracellular MTX. It is also important to note that when DHFR activity was undetectable, minimal UDR incorporation was still found. Figure 3 further shows that when DHFR activity recovered to 5% of control, UDR incorporation recovered to 50% of control, reaching 100% when DHFR activity was 20% of control. Thus, it can be shown that the free MTX, or excess accumulation of MTX, is required to maximally suppress both UDR incorporation into DNA and DHFR activity, more specifically the reduction of DHF to THF.
Methotrexate enters the cell through active transport. A constant amount remains bound to DHFR, and a variable amount of free methotrexate accumulates in the cytosol. The results suggest that free methotrexate is required to maximally suppress deoxyuridine incorporation into DNA and suppress DHFR activity. From Table 2, we see that deoxyuridine incorporation declines from 61% of control at the equivalence point to 0.89% at a methotrexate concentration of 10 millimolar. The equivalence point is where all high-affinity sites are saturated and there is no free methotrexate. From Table 3, we see that DHFR activity drops from 6.09% at the equivalence point to 0% at a methotrexate concentration of 1 millimolar or greater. If only 6% of DHFR activity is sufficient to permit deoxyuridine incorporation to continue at 50% of control levels, this suggests that free methotrexate is necessary for the most effective inhibition of DHFR and subsequent DNA synthesis.
One lingering question we have is whether or not stoichiometric inhibition is the main factor that led to the subsequent inhibition of DNA synthesis. It appears from our data that DHFR-MTX is slowly reversible, indicating that the stoichiometric inhibition is not the only interaction occurring in the intact cell at physiological pH. If the MTX-DHFR association is slowly reversible, kinetic conditions would dictate that maximal inhibition of enzyme activity requires free MTX in the intracellular fluid to keep all the enzyme sites saturated, which was shown in Figure 3 previously. This view is consistent with the fact that there is a sigmoidal relationship between total intracellular MTX and DHFR activity, or UDR incorporation rates. Hence, this challenges the original theory that it's due to stoichiometric inhibition of DHR that led to the subsequent inhibition of DNA synthesis. However, another possible explanation would be that there are several forms of DHFR with different affinities for MTX. Unfortunately, this has not been studied as intensively and remains a subject of speculation, as these DHFR affinities for MTX have not been examined. After comparison of all these theories, the likelihood of slowly reversible binding of MTX to DHFR is the most consistent with the observed high dosage of MTX in certain human neoplasm. Since MTX has low permeability, cells are unable to concentrate MTX intracellularly, making it less likely to achieve free cellular MTX. However, at high doses of MTX, it favors the accumulation of that free MTX. This brings to our final few questions of how high a concentration of MTX must be achieved to reach that dosage. Does it vary with different tumors? We hope that additional studies with different types of tumors will provide answers to those questions. Thank you so much. That's the end of our presentation, and special thanks to Professor Mitra for this semester.